So we're going to talk a little bit about the FDA and biocompatibility. And I'm super excited about this because in my line of work, I've been doing biocomp now for 15 years. And when I first started, we worked off a guideline called the G95 or the Blue Book, okay? And that was in 2002. And for the longest time, up to June of last year, we kept working off of that Blue Book. But it kept on getting less and less relevant. In fact, what happened is we started getting feedback from the FDA saying, we want you to do this. Okay, so we'd write a little note. So the next time we had a sponsor contact us, we would say, this is what the FDA prefers now. So it got to a point where we were just telling people what the FDA has said recently, and we didn't really have any guidance to support it. Then in 2013, the FDA surprised everybody with the draft. And that draft document was how their new approach for biocompatibility. They asked for comments on that draft. And after 630 comments, they came back just June of last year with this new guidance document. And this guidance document is extremely good. I would highly recommend, if you're a geek like me and want to know more about Biocomp, to come back to your hotel tonight and read through the guidance document. Or if you're not a geek like me, I'm going to go through the most important things here, so it's up to you. But I would recommend going through it because it is the most up-to-date philosophy for the FDA and biocompatibility. And let's face it, Biocomp is changing quicker and it's more dynamic than ever. Things are going like crazy as far as what's now accepted, where it's accepted, what's Japan doing, what's Korea doing, what's Europe doing. All these things are changing very quickly, and so it's very important to stay up on those drafts, okay? So we're going to try to stay up very up to date on it. So this is what the guidance document looks like. Okay, perfect, thanks. So this is the top of the guidance document. This is the link, like I said, for tonight for your own enjoyment. You could be able to go back and read through it. But we separated this guidance document into 3 categories, okay, that we're going to cover today. The first category is summary of ideas. These are things that we've seen from the FDA historically, you know, those things we wrote down every time we got one, but now they've put it into the draft or into the guidance document. So we're going to cover those. They're kind of impactful in some cases, okay, so we're going to cover through those. The FDA gives us justification ideas. They do not want you to check box the testing. And this is something that we've seen and heard from all our friends at FDA for the last year and a half. They've really said, please do not just go down and check box these tests. As you notice, I didn't say these are the tests that are required for your device. I said these are the categories that your device has to determine, okay? That means we don't have to test. We might have to test. It depends on the risk of your device and how you mitigate those risks. And we're going to talk about that. So those are the ideas that the FDA has included in the document. And also possible impacts. What things have we not seen before that we're starting to see in this document now? Okay, so that's what this presentation is going to kind of cover, these 3 things. The first thing has to do with fluid and gas path devices. If you have a device that has gas path or fluid path going through it, the FDA recognizes that this contact is indirect patient contact, specifically gas. It used to be gas path devices really didn't consider biocompatibility very much. But now if you have gas going through a device, it is considered like a liquid. It pulls chemicals from those polymers and delivers it to the lungs of the patient. So anything that has fluid or gas going through it is considered indirect patient contact, okay? So that's what we have to consider. That also may be handles, fluid pathways in handles. In the past, when you looked at a handle, it might have a fluid going through it, like some kind of flush channel, and that was not tested because it was in the handle. Now, if you have fluid going through it into the patient, we have to consider that as indirect patient contact. Parallel standards. The FDA actually spells out the parallel standards that they want to follow for certain tests. ASTM, different ISO and OECD guidelines. So it helps us know exactly what test methods we need to do for the specific endpoints. The FDA, and this is something that we should all know by now. In fact, I think a lot of people could just recite this by memory. But the FDA does not approve individual materials. They approve final devices. And if there's one thing you guys can walk away from here, because we get this every day on the phone, is this concept where is just because all your materials have passed biocompatibility, just because all your materials are medical grade or they're USB class 6 or whatever categorization you want to put on it, doesn't mean that that device is safe, okay? In fact, most of the time, we see the same materials come through the door over and over. I can't tell you how many times I've tested polypropylene. It's like thousands, okay? So we know the materials for the most part are safe. And the ones that aren't necessarily safe or have some problems, like for example, silver or latex, things like that, we know what the risk is. So the materials themselves aren't necessarily the issue. What we've seen as the issue is the residuals from the processing. If you're going to fail a test unexpectedly, most of the time that unexpected failure comes from something that's done in that process of that device. A mold release was left behind. A detergent wasn't rinsed all the way. Something happened during the manufacturing process. We've seen people who use baths to rinse things, and maybe that engineer or whoever's running it doesn't change that bath very often. Well, what happens when you rinse something, it builds up into that bath, so that last device in there before it changes now has a bunch of residuals on it that now can have some kind of failure. So these are the kind of things we see. So for this reason, the FDA will not approve a device based off its raw material. So if you just write a justification that this is stainless steel and I don't have to test it, you're not going to get approved to the FDA. You have to address the processing too. So there's two parts of every biocompatibility assessment: materials and processes, okay? The next thing goes in the exact same thing, which is master files. I, once again, get all the time, well, my device is made out of NuSeal silicone, and they have a master file with the FDA. So I don't have to test because it's NuSeal master file silicone. Remember, it's not just about the material, the processing. What are you doing to that silicone that could change the biocompatibility? If your device fails, it's not the end of the world. Remember, these tests are predictive tests for biocompatibility. They're not necessarily saying that you're going to kill someone. Okay? We have devices all the time that fail cytotoxicity. And I've had people call me on the phone that says, great, we spent 3 years, $20 million on this device, and now we have to throw it away because it failed cytotox. My heart crumbles when I hear that, okay? Don't let a $170 test ruin your device. Let's look at it. Let's investigate why it failed. Let's determine if it's a true risk or not. Remember, a lot of these tests are screening tests, trying to determine the risk. And just because you failed doesn't mean that that is a true risk to the patient. We need to determine that. So if you do fail, contact your lab, let's talk through it, determine why you failed, and determine if it's a real risk to the patient or not, okay? And if there's any questions out there, I can get on a roll. I really love this stuff, so if you want to shut me up, just raise your hand, and then we can talk as question and answer. So, permanent contacting devices should only be extracted at 50 degrees Celsius, okay? So when we do biocompatibility, we take a device and we extract it in fluid. We put it into some kind of liquid to get the chemicals that come off. We have different temperatures we can do that at. One of them is 37 degrees Celsius, which is body temperature. Makes sense for a lot of people to extract at body temperature. But if your device is in contact with the body for more than 24 hours or 48 hours, the FDA is going to want to see 50 degrees C, okay? Elevate that temperature to see what happens over a long-term exposure with the device. The only exception to that is if your device changes chemically by its close to its 50 degrees C, okay? So if it's thermal degradation changes or whatever it may be, its glass transition phase is about 60 degrees Celsius, then we don't want to use 50 degrees. We want to use 37 because we don't want to change that device chemically. But most materials do pretty good at 50 degrees C, so we can extract at that temperature. This is one that did change for this new guidance document that we've seen the FDA start to ask for. And it has to do with cytotoxicity. For years, in fact, in the ISO standard today, it says do not extract for more than 24 hours at 37 degrees Celsius. Because, and I know I don't want to get too technical here, but the MDM fluid we extract in has L-glutamine in it. The L-glutamine degrades over time. So the thought is that we're making the test more sensitive the longer we extract it. So just do 24 hours. But the FDA has come out and said if you have a device that's in contact with the body for more than 72 hours, they want you to extract at 37 degrees for 72 hours. And this is for cytotoxicity, which before we never did. So if you've been running cytotoxicity, you've probably always done a 37-degree extract for 24 hours. If your device is in contact with the body for more than 72 hours, you're going to start having to do a long-term extraction. And what that means for you guys is if you passed cytotox before, and now you're doing a 72-hour extraction instead of 24-hour, it's a more sensitive test, and so you can start to see some failures now with the elevated extraction. So we talked, samples should be extracted with 5%. There were some options with that one, so the FDA said only extract with 5% calf serum on the media. Here we go with hemolysis. They, once again, do not follow ISO 10993-4 here, and they say if your device contacts blood, they recommend, doesn't matter how long or what type of blood it contacts, it says you have to do both hemolysis, complement, and thrombogenicity. So if your device contacts blood at whatever, we're talking about circulating blood, not blood in a surgical site, but if it's contacting circulating blood, then we have to test for all those blood endpoints. And that's one that's different. Implantation, intramuscular is preferred over subcutaneous. So those are some of the things that this new guidance document has put out for biocompatibility. This one makes me happy. This is probably my favorite slide in the whole presentation, and I'll tell you why. It helps us get out of testing. And I know that sounds weird coming from a lab, but honestly, a lot of these tests aren't very predictive, in my opinion. And so if we can do a better job predicting safety by doing some other types of evaluations than putting it into an animal, I feel like that's better suited for us as the consumers of these products and for these animals that we test on. The first one is benefit of the device to any potential risk. When I'm doing a risk assessment for a device, this is the first thing I think of. If it causes some irritation, that's not great, right? We don't want to cause irritation. But if this device saves someone's life, then they may deal with some irritation, some short-term irritation to have long-term benefits. So the FDA allows us to evaluate the benefit of these devices to mitigate potential risks, okay? So that's something we use in our risk assessments at Nelson Labs. We can do literature review. We talked about processing, okay? So let's say you do some kind of passivation process, and you're looking at that passivation and rinsing. Well, we can look at literature, how that passivation process is used, and try to determine what the risk is to that device. Okay, it's a stainless steel, passivated. This is something we can justify using literature review. But that goes into what chemistry is now becoming. So if you took in the guidance document and you took a highlighter and you just highlighted the word chemistry, guess how much of the document would be highlighted? Almost half of the document would have some kind of chemical assemblance. The FDA is really starting to push chemical characterization for biocompatibility. I'm going to try to help you understand why it's so important. If this is my medical device, right, and I extract it in this water, and then I put this water into an animal, I just stand back and I go, whew, good, that guinea pig didn't have anything go wrong with it, so that device has to be safe for my daughter or for whoever it is. That doesn't really sell me very much. I know we've used it for decades, and it's really been kind of the way we should do it, but it's just a green or red light test. It doesn't give us any other information except for this guinea pig didn't react, or this rabbit didn't react, right? It doesn't really predict safety except for in guinea pigs and rabbits and mice, okay? Chemistry gives us more information. We do basically the same thing. We take the device, we put it into a liquid, but instead of injecting it into an animal, we now do some chemical techniques to look for compounds, try to determine what's coming off. Oh, I have cyclohexylone. Well, okay, where's that cyclohexylone coming from? How much of it's there? I got a plasticizer in my device. Is that good or bad? How much of that plasticizer, if that patient's going to use it over and over again? My device is used in neonatals. Can this amount of compound affect how a neonatal reacts? Because remember, we're injecting it in full-blown guinea pigs, not neonatals. So there's a lot more information that I can get from my chemistry that helps predict safety accurately than just injecting it into an animal. It's a much better science. Now, that's my standpoint because I'm a scientist and I prefer that. But for you guys, probably thinking, what does chemistry give me as far as turnaround time and costs? Okay? So in this document, the FDA has allowed these 4 endpoints to be replaced by chemistry. They actually publicly state that systemic toxicity, genotoxicity, carcinogenicity, and chronic toxicity can be replaced with chemical characterization. To give you an idea, These four, we're going to take carcinogenicity off the table because even though the FDA will get to this in a minute, is starting to require that endpoint. Carcinogenicity can take about 2 years and cost about $1.2 to $2 million, okay? So if you want to do that with us, my card is here somewhere. I'd be happy to have conversations with you, but don't, because what I'm going to tell you to do is do chemistry, okay? But let's take that endpoint off the table and just look at the other 3 endpoints. Those other 3 endpoints are going to cost you between about $70,000, and it's going to take about 16 weeks to do, okay? Chemistry is going to cost you somewhere, depending on how much chemistry we have to do on the device, somewhere between $13,000 to $20,000. And it's going to take about 4 to 6 weeks to do the chemistry, and a tox assessment about 4 weeks onto that. So right there, you look at the cost and turnaround time savings of a chemical characterization program to an animal program. So we have better science, we have better results, and we save time and money. For me, that's a no-brainer, right? And for that reason, this is the biggest growing biocompatibility section we have. Okay, things are going really good for chemistry. But for that reason, not all devices should go down the chemical route, right? If you look at our new chart up here, only devices that have prolonged or permanent contact with the body have these endpoints. So if you don't have these endpoints, you're kind of stuck doing right now. I'll talk about it in a minute as far as some in vitro options we have out there. But right now, you're stuck doing the biocomp testing. But for the long-term, permanent or prolonged contacting devices, chemistry is a great option. Okay, it's time. The FDA is starting to really actually require it for a lot of devices. Any questions before we move on? Okay. This is another thing that can save you time and money, and it has to do with functionality tests. And I've never really understood why companies have a hard time discussing this internally or connecting this internally, but there's a lot of times that a medical device will go through a lot of functionality tests to make sure that it works right in the joint or that that functions correctly in the surgical site. They make these really complex, large animal studies where they go through all these functionality tests, and then they stop the design, they move to biocompatibility, and they do more animal tests. There are a lot of endpoints that we can put into the functionality test can address biocompatibility. Why not hit 2 birds with 1 stone and do some biocompatibility with the functionality tests? I'd be more than happy to get on the phone with you and kind of work through that protocol to put in biocompatibility, even if you're doing it with somebody else. Just the fact that I don't want to do unnecessary animal testing on the back end. So we can do hemocompatibility, we can do some toxicity elevations, some implant evaluations, all in the functionality tests, okay? So really consider that when you're starting to design a device, is where can you put those biocompatibility endpoints. So predicate tests, predicate samples. This works great if you're changing a device, which happens to be the most of the work I get. I get called all the time saying, okay, my supplier A is out of this polypropylene, I gotta get it from supplier B. What kind of biocompatibility do I have to do? Or I gotta move manufacturing locations from site A to site B. What biocompatibility do I have to do? You can use a lot of the information as a predicate device. Because remember, the FDA is concerned about materials and processing. So if you can show that materials haven't changed and processing hasn't changed, we've evaluated a lot of the endpoints that the FDA concerns about with biocompatibility. So there's ways we can justify out of testing by evaluating these endpoints with a predicate device. If you make a device that's similar to the device you're trying to get to 510K or 510K for, then we can get through a lot of this assessment without having to do testing. Unfortunately, if you don't it's hard to say, my competitor is making the same device out of the same material as mine, so I don't have to test. Because remember, it's not just about materials, it's about processing, and I have not met a competitor that's been willing to hand over their processing to another competitor and say, here, now you don't have to do biocomp, right? So there's things that we still have to address that it's hard to do with a predicate device that you don't know much about. But if you know a lot of information about the predicate device, we can justify out of a lot of testing that way and save you guys time and money. Okay, so here's some possible impact items, the things that you guys probably care most about, right? As far as things going forward. Have we ever considered practitioner use before? I've never really considered practitioner use before, okay? So practitioner, right, these are the nurses and doctors that are using our devices. we've always considered the handles and other things of the devices as non-contacting portions of the device and not required biocompatibility. And for the large part, that's still true. But in the new guidance document, the FDA put in a section about practitioner use. And particularly, they said if they're not wearing protective clothing, so if your device is used without gloves or something like that, where skin of the practitioner is going to contact the device, then we have to evaluate the risk to the practitioner, okay? Which is something new. We've not seen this from the FDA before. I have seen cases here and there, but never as a rule. A good example, we had a device just recently that was a face shield for a surgeon, okay? They submitted it without biocompatibility because it doesn't contact the patient. It's a face shield. But the FDA came back and said, we need to evaluate biocompatibility for the sweatband that the face shield has, because it contacts the forehead of the surgeon. So they contacted me and said, what do we need to do here? So we just had to do biocompatibility assessments on that material, because it contacts the practitioner and it's not covered by any kind of protective wear, right? So if it is going to contact the practitioner without any PPE or any protective gloves or gowns or anything like that, then we have to evaluate biocomp. Luckily, most devices have gloves and things like that that they're using, or the surgeons are using, but still think about that when you're thinking about it. Make risk-based decisions. This is big. We have to start thinking about these biocompatibility as a risk to the patients, not just a checkbox. I know I talked about this before, but it's an important key to take home. Every time I talk to the FDA, they say, please let them look at the risk of the device and not the checkbox. So I have to put it in here twice, okay? So please evaluate the risks of these medical devices to the patient. They do encourage the use of master files, but in this new document, they actually specifically give guidance on what a master file should include. In fact, they actually have a section, an appendix in the document, where they list what they want to see in a master file. So if you're thinking about submitting a master file to the FDA, it might be good to look at this section to determine what you need to put in that master file to let the FDA feel like they could use it more effectively. Because remember, they won't accept a device on a master file alone, but they're looking to add some more information in those master files to be able to feel more secure about those materials and the biocompatibility. Okay, so here's one of the first things that I've seen is different, which is we call a biological evaluation plan. But in this document, the FDA says, we want you guys to develop a risk-based plan before you start testing. Submit that plan to them, either formally or informally, to get feedback. Now, this is a really good way to try to get out of some testing, or if you have a very complicated device, try to get feedback from the FDA. So we do something called a biological evaluation plan, or BEP, but it's really just a risk-based plan. So what you do is you look at your device, you look at the materials, you look at the processing, you evaluate the risk to the patient, and then you come up with a plan on how to mitigate that risk. And it may be chemistry, it may be a risk assessment in the document itself about why you don't need testing, all these different endpoints, and then you can give that plan to the FDA. The FDA has been very good about responding to those plans and giving constructive feedback on what is needed. In fact, I saw a plan just 2 weeks ago that told them they were doing too much testing, which I almost dropped to the floor because I don't think I've ever seen that before from the FDA. But they said, you don't really have to do genotoxicity here. We don't think the device has that risk, so you can cross that out. And I'm like, well, this does work, okay? So think about coming up with a plan, especially if it's a complicated device, okay? And submitting that to the FDA. If you are declaring conformity, you have to provide that testing was done before on that device. Once again, easily done with the device you make yourself, not so easily done with before. And this is the reason why the FDA put this in here, is because a lot of the testing requirements has changed. We talked about cytotoxicity changing on its extractions. There's some things in the test methods that have changed. So the FDA wants to make sure that the predicate device that you're claiming has the similar testing as your device is going to have to make that kind of declaration. And then at the end, and they actually put this in an appendix for you to review, but they want you to deliver a report, okay? I think the reason they have this in here is I think there was too many times where someone just gave a bunch of reports to the FDA and said, here's my biocompatibility, and just put about 500 pages of Nelson Labs reports on their desk. I think that probably happened a couple times. So what this report is, is actually just an answer to your plan, okay? So your plan talks about the risk. Your report is just a report that says, this is the risk of my device. This is what I did to mitigate the risk. This is the results from my testing that I did. And in conclusion, we feel like this device is safe for use based on if its benefit to the patient. And then you can actually have appendices with the reports behind it, but the FDA tends to really enjoy that better than just a bunch of papers, okay? And in attachment C, they give examples of what some reports could look like, so you can have an idea of what they're looking for to submit to the FDA. Okay, so we talked about cytotox samples may be extracted 72 hours. That's a possible impact. If you have to do complement activation, they used to require 2 proteins, which is a SC5 beta 9 and a C3A. Now they're just requiring C3A, okay? So it saves you guys money because we don't require both proteins for complement activation. But they did add about pyrogenicity. Do you guys know what pyrogenicity is? It's fever, something that causes a fever, okay? So there's really 2 types of things that cause fever. We have something that's called endotoxin that's found in gram-negative bacteria, okay? And that's looked at by using an LAL test, and then, or bacterial endotoxin test. And then we have everything else that could cause a fever. And we kind of screen for that using a rabbit test. Historically, you only ran the bacterial endotoxin test or the LAL test for devices that contact like the eye or the cerebral fluid or the lymphatic system or the blood, the circulatory system, and that's it. And then you ran that rabbit test on everything else. But in this document, and there's been another guidance document released for orthopedics, the FDA is requiring all implants to have LAL testing done on them. So no matter where you're located, if you're considered an implant, the FDA wants you to do a bacterial endotoxin or an LAL test on that implant. And this is a big impact to orthopedic industry. The LAL test is cheap and quick. It's not the test, it's the samples. These orthopedic samples are not cheap, and they take a long time to manufacture. So now we have to submit more samples for testing. So this was a big impact to the orthopedic industry as far as pyrogenicity. Okay, so the other thing we talked a little bit about is carcinogenicity. Previously, the FDA kind of allowed us to use genotoxicity to justify that carcinogenicity is not needed. Now remember, carcinogenicity is that test that takes about 1 year to 2 years and costs a couple $1 million, right? So no one did that test. Everybody kind of justified it by doing genotoxicity and then just saying we don't really need to do it. The FDA now says, and it's true, it's totally scientific true, that genotox cannot justify out of carcinogenicity. And they're requiring if you have carcinogenicity marked on this slide rule, they're requiring you to address that endpoint. Now don't freak out. You don't have to do that test that costs a ton of money. Remember, chemistry is one of those ways we can address carcinogenicity. So if you have carcinogenicity marked on this chart, you're almost stuck doing chemistry nowadays, okay? It's a way for us to justify out of that endpoint along with the other ones. So we talked about chemical characterization already. There are some devices in this document that the FDA is actually requiring chemical characterization for. Gas path and fluid path devices, the FDA is asking for chemical characterization. And then anything that's considered a novel or new material. If it's a new material not used in its indication, or if it's an air or gas path device, the FDA is requiring chemistry, okay? Now, the last thing we're going to talk about as possible impacts is changes in table A.1. And this one possibly can be huge. This is this slide rule right here. So as you guys may notice or been familiar with, this is the table that everybody, if you look up biocompatibility on Google, this is probably one of the first images that pop up, right? So what you want to do is this is how you determine your risk. The FDA added 3 new endpoints. They added material-mediated pyrogen, which was not included in the original, and it's still not included in ISO 10993. It's just assumed that it's an acute systemic toxicity. They added reproductive developmental toxicology and degradation. Then they also put in here, and this is the biggest impact item. Oops, sorry, wrong way. At the very bottom there, they say that all circles have to be evaluated like all other endpoints. And this goes back to remembering that these X's are not tests that you have to run. They're endpoints that you need to evaluate. They've added all the circles, the FDA only. If you're doing to Europe and using just ISO, you don't have to worry about the circles. But if you're submitting to the FDA, all these circles now have to be evaluated just like the X's do. Okay, and this is a change from the old document. So when we go through this, you start looking at some of these circles and you start looking them on chronic and carcinogenicity endpoints. This is where we start having to do some chemistry testing to evaluate those. Okay, so when you look at this chart right here, when you get my slide rule afterwards, remember that we don't use circles, we use diamonds because I don't know, I think diamonds look better, I guess, happier. But these diamonds are the same endpoints as the circles. So if you have diamonds, the FDA is going to require you to evaluate those endpoints. Okay? So here's some additional information. I do want to mention, we talked a lot about justifying out of biocompatibility tests with animal tests using the chemistry. We are looking at also justifying out of some of those endpoints by using new in vitro methods. In fact, we're very close to a new in vitro irritation test. It's right now in the biostatistician. So it was an ISO ran round robin test that we did it at 13 different labs. And then all these results are put into a biostatistician. He's actually working on his report right now. He should have it done any day now. We'll submit that to the FDA. And as long as everything looks good, and by the way, it looks pretty good to me, then the FDA is going to start accepting an in vitro irritation test. So if you look back at these endpoints, you got the big three, right? Cytotox, sensitization, and irritation, which every device has to evaluate. Cytotox is an in vitro. Irritation and sensitization are animal tests. We're close to getting in vitro alternatives to those two tests, which means for you, once again, no more animals, but they're also cheaper and quicker than the animal tests. So my goal, and this is what I want to do before I die, right? I want to make it so that you guys will only have to run an implantation and some other large animal tests or functionality tests, and everything else will be in vitro or chemistry based. Okay, so we're getting there slowly. There's labs out there that are looking to take away other animal tests like the in vivo dog thrombo. They're looking, actually APS right here, they're working on getting that as an in vitro alternative. So there's a lot of efforts to reduce the amount of animal testing we're doing. Also reduces time and money for you guys. So I had those things up here that you can reference for those endpoints. Like I said, if you want the presentation, just come bring me a card and I'll be happy to do so. Any final questions today? Okay, well, oh yes. So, good question. Let me just clarify. She said for implant devices. So there's one endpoint that chemical characterization can't help you with. It can't help you out of implant itself. But if you have an implant that has less than 24-hour contact with the body, I would just do the testing. Because if you look on this, then if you look at an implant for less than 24 hours, see how cool this slide rule is? You guys should all come get one. So you're only doing the big three plus systemic toxicity and material-mediated pyrogen. So even though technically you could use chemistry to get out of the systemic toxicity, that test is really cheap and quick. So it doesn't make a whole lot of financial sense to do chemistry to get it out. So for that device, I would just do the testing. But if you had longer than 72 or 24 hours, if you were prolonged, now you're basically doing everything. And that's where chemistry can help you justify. Good question, though. Any other questions before we move on? Okay, well, come grab a slide rule, and if you do have any additional questions, I'm in booth 2930 upstairs, so you can come see me. Thank you.